All right. So today may be a little bit different than usual because I'm really not going to play around with the terminal or RStudio. Today is largely going to be dedicated to using servers, using websites. So that being said, here's kind of a disclaimer. If this gets laggy, just let me know. If it gets a little too laggy, let me know, and I'll kill it and ultimately just upload a video this afternoon as opposed to doing this live if it gets a little too laggy. So keep that in mind as we're going through today's stuff. So last week I ended off with more or less exporting results, or we ended up plotting some of the differentially expressed genes. Right, at least a couple of different ways we can plot them out. Heat maps, MA plots, volcano plots, etc. But I've exported all of that stuff out already. Like at least the main file. So for example, like the DE-seq water 15 PSU. So I'm just going to quickly open this one up in Excel. And so today will be a decent amount of Excel work. I mean, not a whole lot amount, but at least a decent amount. All right, so this is generally the raw output of like results of DE-seq. So it tells us, which genes are differentially expressed with the log fold change, and if it was significant, at least if a p-value was significant, right? But this really doesn't tell us a whole lot about the functions of these genes, right? LiPo means nothing to me by itself. HRG means nothing to me. But we can kind of figure out if there are genes involved in a specific function that seem to be affected by the experimental conditions. Right? Again, we have turtles sitting in water, pure water for 24 hours, and turtles sitting in salt water for 24 hours. Extract the livers, see what genes are expressed and how it changes. All right. So moving from there, I put a link to this website in the description, but it's just gene ontology, aka it's also called Go, gene ontology. And so this is kind of like a go-to way to at least figure out general functions of a list of proteins. Really even just one protein. Again, if this gets laggy, just let me know and I'll just kill it and make a video. So gene ontology is like if we figure out a function, say in mouse, of a specific gene, and it seems to be involved in like the electron transport chain, then we'll apply a Go term to that specific gene. Well, like, at least it'll probably have a Go term of like mitochondria and a Go term of ETC. Something along those lines. And furthermore, if there are like specific domains within a known protein that we know about, then we can also apply another function. So for example, if we see a helix turn helix domain or a zinc finger domain within a protein, well, it implies it's going to be DNA binding and maybe a transcription factor or a polymerase. Right? It's going to be involved in expression. Right? And so we can at least gather these general terms and apply them to specific proteins. And so ontology, at least if we look at the gene ontology overview, each gene will ultimately get terms devoted to 3 different categories that are also kind of overlapping: molecular function, cellular component, and biological process. So For example, if we have a gene that's specifically involved in the ETC, then its cellular component is probably going to be mitochondria. Molecular function may be electron transport, and biological process may be ETC. Something vaguely along those lines. And so we often issue these terms, so we just call it a Go term, gene ontology term. And so like by default, like there will just be a specific number. So like go and then some number. Go colon some number. Right, the Go term. And so a lot of Go terms are nested. Like even if we look at this general example, there might be a Go term for metabolic process. But even within metabolic process, right, that's just a general overall term. But within that one idea of metabolic process, we may see a biosynthetic process, a small molecule metabolic process, maybe a lipid metabolic process, a carbohydrate metabolic process, etc., etc., etc. So these terms may get broken down. Or, for example, if we have, monosaccharide biosynthetic process, right? Whatever that gene is involved in either the construction or breakdown of a carbohydrate, it's going to get monosaccharide carbohydrate biosynthetic process. But that's also a part of the metabolic process, right? So these Go terms get kind of nested generally, right? Even in the most descriptive Go term is still going to be kind of vague and nested within a general larger process or more generic process. And so we can at least figure out, so if we have a whole list of genes, we may be able to identify all the Go terms listed to those genes. Right, that's kind of the nice thing about gene ontology is gene ontology and UniProt are reasonably well linked. So if I give, let's see, I don't even know if I can do this with Go, right, the raw website. But say, for example, if we can certainly do this in UniProt. So let's do this in UniProt. So UniProt. For example, if I have MCRS1, stop. Let's see if I can get any specific Go terms for MCRS1. Okay, so we do have these Go terms. So these aren't going to be like the Go symbols, but we get the Go words. So we get a molecular function regardless of what the name is. Oh gosh, microspheral protein 1. Fine. So it's poly-G binding, poly-U binding, telomerase inhibitory activity, right? Go terms. Sometimes not super helpful, sometimes really helpful. And so we only get Go terms for the function and the biological process, and it's largely cytoplasmic, right? So you're going to find this thing in the cytoplasm. Whatever. So again, like each gene has a whole list of Go terms. And so if we give like gene ontology, like a reference database of genes, it's going to find all of its Go terms. And if we have a list of, say, upregulated genes, we can also give it that list and see if there's a difference in Go terms between the reference list and the differentially expressed list. Right, so that's how we can use these Go terms. Let's see what else we can do. And so we're going to do that. And so we're going to run this thing called Panther. So Panther kind of drives this. It's kind of this internal module, this internal analysis within gene ontology. But Panther does have its own generic website, right? pantherdb.org. And so this is where we have a little bit more control over gene ontology stuff. All right, so we have our database, or we have all of our Go terms. So first of all, let's get all of these genes out into a text file, because working with text file in gene ontology or Panther is way easier. So I'm going to copy all this entire row or entire column and throw it into a text file. That'll do. And get rid of blank lines, because you never know what a blank line is going to do. Most of the time, nothing, but sometimes it does something. So I'm going to save. I'm just going to. I have this now. I have all the genes that we found, that we identified in all of our liver tissues, about 11,000 genes. So I'm just going to save this as all genes.txt. No. All right, now I'm going to use Excel. So we can always do this in R if we so wanted to, but it's a little tedious. If you know, sometimes, very rarely, it's faster to do things in Excel than it is in a programming language. And filtering, I think, is one of those things. So I'm going to do some filtering stuff. And so I want the differentially expressed genes now. So I have a list of all genes. Now I want to make a list of the differentially expressed genes. So I'm going to set a number filter to my p-adjusted value that is less than or equal to 0.05. And that should give me all my differentially expressed genes. It's 200 some odd, right? 270. So I'm going to copy these genes, throw that into a text file, and just call it different, like diff.exp. Right, I'm not too concerned about whether it's higher expressed in salt water or lower expressed in salt water. Not there yet. Just want to deal with the differentially expressed genes. All right. So Panther gets a little silly. I'm not gonna lie. Panther gets a little bit silly whenever we're trying to do a specific test that is the statistical overrepresentation test. And so with a statistical overrepresentation test, that's what we're looking for. We're looking for Go terms that seem to be more common in our differentially expressed genes than in the data set as a whole, right? So we're looking for an overrepresentation of Go terms. So for one, I'm going to supply my text file that's the differentially expressed ones. I'm selecting human because human is one of the best annotated, right? There's not a turtle in this list, and probably the most closely related thing to a turtle in here is probably going to be a bird. But even still, like, the best annotations we're going to get is for the human. So I'm just going to assume a human data set. Not uncommon to do in bioinformatics. And so, fine. And so it should take me to another page where I can upload a reference data set. And so that's going to be my all genes. choose file, all genes, human, gene transcripts, alternative ID, upload list. Okay, so now on my reference list, that's all genes. My analyzed genes, that's the differentially expressed ones. And we're leaving everything else by default. So it's going to be a Fisher's test, and we're correcting for multiple tests, right? Because we're doing thousands of tests. And let's see what happens. And so the first test I'm running is the biological process, right? So again, we have terms for biological process, molecular function, and cell component, right? I'm not worried about Go Slims, not worried about Slims yet, like a reduced data set. I'm kind of going against the entire complete data set. All right, and so what is returned is a list of terms or functions that seem to be enriched or more common to the differentially expressed genes than we'd expect by chance. And all of these terms are more common than we'd expect. And so again, we see like the main terms and then the nested terms, right? Regulation of coagulation is a term. Positive regulation is a nested term. Positive regulation of blood coagulation. So these genes seem to be involved in blood coagulation, at least to some degree. That seems to be a general function. Is a lot of these terms are linked to homeostasis and coagulation of blood and response to healing. Let's see. We also see a little bit of metabolic process, NAD metabolic process, right? Right, that's more or less an electron transfer molecule, NAD, right? NAD plus NADH. Antimicrobial response, so maybe a little functions and maybe a regulation. Response to microbes seems to be affected a little bit. Pregnancy, that's a small term, but nonetheless, it would be enriched. And a couple others, right? Still kind of vague, but we have somewhat of a general idea of what seems to be affected, right? The types of genes that seem to be affected when we put a turtle in salt water for a little too long. And so again, like for all these terms, so like we do have these several columns, right? So we have the genes, all genes.txt, that's the reference file, and more or less how many genes had this term. Right, so 22 genes out of 11,000 had this positive regulation of blood coagulation term attached to it. And if we understand that, so 22 out of 11,000, you would expect less than really just a few genes. So if we can think of it as a ratio, we have 22 genes out of 11,000, right? That would be a background rate for that Go term. 22 genes out of 11,000, going to be a really, really small number. And then whenever we compare like the genes that have that term in our differentially expressed list, it may be higher or lower. In this case, it's substantially higher. So we're expecting less than one gene out of whatever we had, 270. Less than one should have been devoted to positive regulation of blood coagulation, given our reference. But in fact, 6 genes. 6 genes were linked to this, positive regulation of blood coagulation. So that would be an enrichment. We're expecting 1 or less than 1, but we see 6. So that would be, that term is more common than we'd expect by chance among the differentially expressed genes. Not by a lot, but a little bit. But nonetheless, we also see other terms related to this. Like we see 12 genes with regulation of coagulation, and 63 are in the reference. So again, we'd expect about 1.5 genes out of 270 to have this term, but in fact 12 do. Right, so that's how we can kind of figure out this, if there's an enrichment for specific terms. As by yeah. And again, positive, negative, what direction the fold enrichment is. Like, do we have a more commonly expected or less commonly than expected? In this case, it's all going to be more commonly than expected in the fold enrichment. And FDR is just a same thing as we saw in DE-seq. It's just a correction, a false discovery rate, or a correction for doing multiple tests. But either way, so again, we can run this test multiple times. This was just biological process. I think honestly, the easiest go category to understand at our level is going to be the cellular component, because we know the components of a cell, right? Mitochondria, nucleus, cytoplasm, etc. And so we can just see if any component of a cell seems to be more affected than others. Cell membrane, outside the cell, and the nucleus, mitochondria. Where do these proteins that are differentially expressed localize, and is there enrichment for it? And so two terms seem to be more common: blood microparticle and extracellular exosome. So extracellular, right? So we see some extracellular stuff, proteins that are going to be expressed outside of a cell, or going to have a function outside the cell. And the kind of cells or kind of proteins that do this are going to be involved in coagulation. So in this regard, this kind of makes sense, right? We have blood coagulating proteins being affected, and they're expressed outside of a cell. And also, blood microparticle is kind of in the same thing. It's like a vesicle, right? It's a vesicle outside of a red blood cell, and probably going to be involved with coagulation of some degree. Now, so this is kind of interesting. Like, right? We got a transcriptome from a liver, but we keep seeing a lot of these blood proteins pop up, or at least coagulation proteins pop up. I don't know. So that's a bit interesting. But that seems to be what we find more often than anything else, is related to coagulation. Molecular function is not the easiest one to understand by far, because that's going to get really, really detailed in molecular biology. But we can still just take a look and see if we understand anything that pops out. Yeah, so not helpful at all, at least for me. Somebody who's probably a bigger nerd with this probably knows more. But endopeptidase, regulatory. Not the most helpful term, but $1 says if we click on this, it will give us some description, and $1 says it's related to coagulation to some degree. Endopeptidases. So an endopeptidase, like the thing, the nice thing about biology is if you start understanding the parts of the words, you can kind of figure out what a protein is doing. So like endo is going to be like within, peptide is going to be a protein, and ace probably would suggest it's going to cut amino acids, right? So it's going to cut down proteins. Hydrolysis, or it's going to build proteins. If it catalyzes the hydrolysis, it's probably going to break proteins down a little bit. But that's as good as we can get, at least in this regard, of trying to identify what this term actually means. A little tricky, but sometimes helpful, sometimes not so helpful. Let's see if I can do one more of a pathway. Let's see if we can analyze pathways, specific pathways that are differentially involved. I haven't run this yet, so I honestly have no idea what's about to happen. Blood coagulation. That's the only pathway that seems to be involved. Kind of a no surprise given everything else. But that seems to be affected. All right, and let's kind of do this one more way and break this down into genes that are lower expressed in salt water and genes that are higher expressed in salt water. Right, so let's just break this list down a little bit into those two components. Is it higher expressed or is it lower expressed? So I'm going to go back home and change my list a little bit. I guess I need to make a new list too. All right, so the log fold change. We can manipulate this category as well. So the log fold change. If this number is positive, the expression is lower in saltwater turtles. So if I set this, if I do a number filter and greater than 0, these are going to be the genes that are lower expressed in salt water. So they will be down regulated. So I'm gonna copy all these and put them into a file called down. Or down express. And I'll make another one that's the up expressed, like if the genes are higher expressed in salt water. And so that log fold change is going to be less than 0. Okay, so I'll copy this list. And call it up. So now we have down and up. All right, remember, like most of the time, if we're seeing a differentially expressed gene, most of them are less expressed in saltwater turtles than freshwater. But again, let's just play around with this. Let's choose our. Let's go down first. You know what? Let's go up first. Let's go up. See what genes are up expressed. See if that change, if there's any change there. Biological process. Right. So no, I still need to add my all genes. Let me add my all genes. Okay, up expressed, all expressed, and biological process. Okay. See if anything pops out. Nothing popped out. And so that's annoying, but not entirely surprising, because we're only talking about 77 genes. And so to find anything relevant among 77 out of 11,000 is going to be a little tricky. But I guess if I do a no correction, if I just list like no correction, maybe something will all be trending on significant that's of relevance. Probably not. Always gets a little risky when we filter for no correction. All right, we're more likely to see false negatives. Yeah, blah. Inflammatory response to stress, stimulus, immune system. But again, not very well, not super beyond expected enrichments. And again, it's probably largely because of it's a low sample size, right? 70 genes out of 11,000, right? It's hard to figure out any differences. So let me do this one more time for the downregulated. See if there's more in the downregulated stuff. Let's see if we can get something to pop out there. Right? If I don't select a reference file, it's just going to assume my data set or my reference is the entirety of the human genome, right? So all 20,000 genes in the human genome, right? But that's definitely biased, right? We don't see all 20,000 genes expressed. We only see 10,000. So we want to know what's different given the 10,000. But the default would be the entirety of the human genome. Okay, yeah. All right, so yeah, so this is what's driving blood coagulation, is the down expressed genes. So genes that would be involved in healing and coagulation and things like that are less expressed in saltwater turtles than in the freshwater turtles, right? So that could be a problem for them turtles. Again, if I just do a pathway, $1 says if I do the Panther pathway, it's probably just going to be blood coagulation, if anything. Right, not super bioinformatic-y, but definitely like this is going down a rabbit hole. You definitely have to spend a lot of time going down this annotation go thing. Yeah, blood coagulation. Right, so that could be a problem for turtles. If they're sitting in salt water for a little too long, they may not be as good at healing themselves as they should be because these genes are affected. That seems to be a response. One of the responses. Neat, right? And so all of this stuff can be exported if we want to export it as an Excel table and like plot out the fold enrichment or these terms or anything like that. We certainly can. But we have an idea of what's differentially expressed or what's more likely to be differentially expressed. And it seems to be stuff involved in coagulation, healing, that kind of feature. So I think that's all. I think that's all I'm going to do for today. Again, just kind of figuring out what's differentially expressed and relevant and things like that, and kind of neat. Sometimes I don't know if this really makes biology make a lot of sense, but it definitely makes more sense than some of the stuff that I've tried before. I've certainly done DE-seq before and returned nothing, right? Very, very irritating when we don't find differentially expressed genes. That gets a little bit harder to explain. But nonetheless, so I'm going to call it there for today. And then, honestly, yeah, we'll see what happens. We'll see what happens on Wednesday. Otherwise, I'll talk to you all then.